In this video, we're going to be talking about chromatin structure, how the DNA exists within a cell. And the quick answer here is that DNA doesn't just exist in a naked state within the nucleus. It is packaged up by lots of different proteins. And so when we talk about chromatin, what we're actually talking about is the duplex DNA molecule, right? So that's the naked DNA that is wrapped up around and associated with many different proteins. So if you were to look at a chromosome, now this is a chromosome, this is a duplicated chromosome, exists in the metaphase stage of mitosis, and so this is just before a cell is going to divide into two. When you look at a chromosome like this, about 50% of that chromosome is made up of DNA and about the other 50% is protein. So this chromatin, this DNA protein complex, is quite important for several main functions here. It's important to compact that DNA into the cell. It protects DNA from damage. It ensures that DNA properly divides during mitosis and meiosis, and it provides organization for gene regulation. And we're going to get, again, this way that chromatin, right, the interaction between DNA and proteins, can sort of organize gene regulation or provide a way for gene regulation to occur will be important for the journal article that you are reading for this course. There are linear chromosomes. Those are the ones that are in our cells. And those linear chromosomes, there's a very special problem that occurs at the end, each end here. Remember, this is one double-stranded DNA. This is another double-stranded DNA molecule because this is a replicated chromosome. In fact, this chromosome doesn't look at all like this 99.9% of the time within a cell. So each end of that double-stranded DNA molecule has to be protected, and that has also some problems with DNA replication that we'll get into much later in the course. Circular chromosomes, they don't have to worry about their ends because they're circular, but there is a problem with daughter chromosomes that sort of get topologically linked at the end of DNA replication. So each one of the types of chromosomes does present a specific problem that the cell needs to have a solution for. So let's talk about the most primary level of chromatin structure, and that is the nucleosome, which is a unit of DNA wrapped around a histone octamer core. Now, these octamer cores, these nucleosomes, these proteins are called histones, and there are 1, 2, 3, 4, 5 different histones. Now, H1 is a little bit different. It exists outside of that octamer core. The octamer core is made up of 2 H2As, 2 H2Bs, 2 H3s, and 2 H4s. These proteins are extremely conserved. If you were to compare a H4, histone H4 between humans and yeast, you'd only see 8 out of 102 amino acids that are different between the two. And all of these histones have an extremely high percentage of lysines and arginines. Now, these are the negatively charged, these are the positively charged amino acids. Why would that be the case? They have strong association with the DNA. The DNA has a net negative charge, so of course these must have a net positive charge. So the histidine positive charge stabilizes the negative charges on the DNA itself. This is what the octamer core looks like. It is a H3H4 tetramer and 2 H2A, H2B dimers. And we'll look a little bit more closely at what I mean by tetramers and dimers in a minute. So there's a total of 8 proteins here, 2 copies of each of 4 histone proteins. And then the DNA is wound around that a little less than 2 times, about 146 base pairs. And so this winding of DNA around our octamer core is one nucleosome unit. And if we look at a whole bunch of nucleosomes strung together, what we can see is each one is linked together by what's called linker DNA. That's DNA that is not involved, wrapped around the nucleosome. And it's about 54 base pairs. It can be bigger or smaller in different organisms, but in general, on average, it's about 54 base pairs of DNA that links one nucleosome to the other nucleosome. And if you were to take a very detailed look here in electron microscopy, you would see that these nucleosomes look like beads on a string. Now, one thing that these previous images did not give you a feel for is that each one of these 4 histone proteins are rather compact, but they all have an N-terminal tail. A certain number of amino acids that are not involved in a beta-pleated sheet or an alpha helix, they are structureless. And so they just sort of extend out of the nucleosome, free floating, just like this. So these are the N-terminal tails of the various histones. And this is super important because what they are substrates to various modifying enzymes that will add covalent modifications to these tails, like methyl groups or phosphate groups or acetyl groups. All sorts of different modifications can occur to these N-terminal tails, and those modifications do several things. First, those modifications will affect how adjacent nucleosomes will interact with one another. So what this can do is this can close that chromatin up and make it very hard for any genes to be expressed, or it can open the chromatin up and allow for gene expression to occur. In addition, the modifications on these various histone tails can also act as like recruiting platforms, like codes to which proteins will be attracted to. So maybe if this H4 N-terminal tail, if it's not phosphorylated, nothing's going to bind to it. But if it becomes phosphorylated, that may attract very specific proteins that will come, bind to that H4, and then start manipulating the DNA in specific ways. And we are going to talk a ton about this within your paper and when we get to regulation of gene expression a little bit later. All right, so we had a problem in that we learned about topoisomerases and the fact that all they can do is relax supercoiled DNA. So then how is it that all of eukaryotic DNA is underwound and therefore negatively supercoiled? Right? We don't have a topoisomerase that can incorporate supercoils, so how do we get supercoils incorporated? And here's the answer. Let's just, I know our chromosomes are linear, but let's look at just a plasmid DNA, circular DNA. And this is what happens, is that DNA, the cell forces it to wrap, right, negatively around the histone cores. Okay, and those are negative supercoils. That's negative rise. Now remember, we did not break that DNA. We did not allow for the linking number to be changed. And so has the net linking number been changed? No, it has not, because we have not done anything to break that DNA. Which means for every negative supercoil that we have put around these histones, there is going to be a reactive positive supercoil elsewhere within that chromosomes. So that the net effect here is 0, is that we haven't done anything. We have some negative rive, we have some positive rive, so that cancels each other out. That's no rive at all. And that is exactly what would happen if we don't change the linking number. Okay, but we do have this. We've got this positive rive here that occurs at this portion of the DNA. And that positive ride can be a substrate for our topoisomerases that, what do? They relax supercoils. They relax DNA. So now we have a substrate for our endogenous type topoisomerases to cut, relieve those positive supercoils, and now all that leaves us here are these negative supercoils that we forced into the DNA. And this is permanent. These supercoils are permanent. In this particular position here, if we remove these supercoils, these wraps around these histones, then that DNA would go boom, right back into the relaxed state. Because negative writhe and positive writhe is no writhe at all. But here, we've now changed the linking number such that now the linking number is less than LK naught. So that when we remove everything, we have a situation where the twist is going to be greater than the linking number, which means the writhe has to be negative, and we now have a permanent negative supercoils. And that is how supercoiling is formed. That is how underwinding occurs within eukaryotic cells. Now let's get to a little bit of detail for nucleosome assembly. So what happens here is we get one H4H3 dimer that then interacts with another H3H4 dimer, causing an H3H4 tetramer. That's what the DNA winds around and then recruits two H2A, H2B dimers. And now we get the octamer core there. And notice, look at all the 1, 2, 3, 4, 5, 6, 7, 8 N-terminal tails hanging out of that core, just waiting to be modified by various enzymes that affect the structure of that chromatin and recruit proteins to specific nucleosomes. Now, that is the most basic form of chromatin. Okay? If we add H1 into the mix, what that does is so H1 exists at the outer portion of each one of these nucleosomes, and it kind of regulates a little bit more the entrance and exit of the DNA that is wrapped around that. So without H1, our beads on a string look basically like beads on a string. In the presence of H1, we now get what's called a 30 nanometer fiber. Okay, this is sort of an organized structure of nucleosomes. Then those 30 nanometer fibers begin to coil up on themselves and interact with a nuclear scaffold that is made up of a bunch of different proteins, including topoisomerases and what are called structural maintenance of chromosome proteins, or SMC proteins. This includes cohesins and condensins. So if we take a little bit closer look at a chromosome that is really beginning to condense, that it's getting ready to prepare for mitosis, to divide, we see this. So here we have we have a sister chromatid here on the left, sister chromatid here on the right. Each one of these are double-stranded DNA, so double-stranded DNA here, and double-stranded DNA here in the blue, and double-stranded DNA on the light blue. So they are replicates of each other, but they're still associated with each other until that cell divides. And how do they remain associated? By cohesin proteins. So these cohesin proteins, right here, right around the centromere, they topologically link the two sister chromatids together. And those cohesin proteins are only going to break when that cell is ready to divide, and the two sister chromatids should divide during anaphase. If we look at the chromosomes a little bit more carefully that are really condensed, we have these condensin proteins. Now, you don't need to know the details about these proteins, but just know that, again, they are proteins necessary for the continued compaction of a chromosome during mitosis.